Hello everyone, today we are talking about eukaryotic and prokaryotic cells. So before we get going on the cells, I want to talk a little bit about how we know what we know about cells, which is by using microscopes. So the one on the far left is an optical microscope using a light. What happens is the light shines through the specimen, through a number of lenses and into your eye. So you actually get transmission, you get light transmitted through the cell, and that's what you can see when you look through this microscope. If you look at the top picture of the onion cells, you can see that the light part where the cytoplasm is, that's where the light is transmitted through easily. The dark blob, which is the nucleus, that absorbs a lot more of the light, and that's why you see it as being black. A decent light microscope can magnify to around about 1,000 times usefully. The TEM, the middle one, that's transmission electron microscope, and that does a very similar thing. Instead of using light, it uses electron beams. An electron beam is not restricted by the wavelength of light, so it can see much better. A transmission electron microscope works in very much the same way, so the electrons go through the specimen, so you see a slice through it, just like you do with a light microscope, but under maximum magnification. You're looking at something like 500,000 times magnification here. Now, SEM, the scanning electron microscope, this is slightly different. This one fires electrons down at the specimen, they bounce off the specimen and they hit detectors all around. That's sent to a computer which does a sort of surface rendering. So with a scanning electron microscope, you actually see the outside of the specimen. The bottom picture here is some pollen grains, and you can see what I mean. They have the outside of the specimen showing. So optical transmission and scanning. A scanning will do about 30,000 times magnification. So those are the three main types of microscope that are used to determine what cells do, what they look like inside and out. Now let's get on with it then. Prokaryote. Pro means before and karyote means nut or kernel, but what that really refers to is the nucleus. So prokaryote means before nucleus. So there's no nucleus, there's no big lump in the middle, no big membrane bound nucleus. So the bacterial DNA, the chromosomes, are free to float around in the cytoplasm. There is also these things called plasmids. And these plasmids are little circles of DNA. And these circles of DNA carry some really important information. And they can do something quite special, these bacteria. They can line up next to each other and send down this little cytoplasmic pathway. It's called conjugation. And they can pass these bacterial plasmids between the cells. Now this is pretty good for them, but not so great for us if you're talking about things like antibiotic resistance genes being transmitted from one bacteria to the next. So rather than having this single membrane bound nucleus, they have chromosomes floating around and they have this plasmid DNA. Prokaryotes, we've got the bacteria and we've got the archaea. Now you're probably familiar with the bacteria. The archaea are usually single-celled and they're extremophiles. What this means is they like extreme environments. Phile means love, philic, love, extreme. So you get thermophiles, which love the heat. You get halophiles, which are very salt tolerant. So these archaea, ancient single-celled creatures that like extreme environments where other creatures can't exist. Now the average size of most bacteria is about 0.2 to 2 micrometers. A micrometer is a millionth of a meter, or a thousandth of a millimeter. So this picture here shows you the difference. In the blue circle we have a bacteria about 0.2 to 2 micrometers, and then we've got a eukaryotic human cell, which is a neutrophil. It's about 100 micrometers. So we're talking about an order or two of magnitude bigger. So eukaryotic cells are much larger than prokaryotic cells. So prokaryotes have no membrane-bound nucleus, but they also have no membrane-bound organelles of any sort. So for example, they have no mitochondria, they have no Golgi apparatus, they have no endoplasmic reticulum. So it's not just a nucleus, it's no membrane-bound organelles of any sort. So to summarise, prokaryotes are the bacteria and the archaea. They're ancient, they're simple, they're tiny, and they have no membrane-bound organelles like a nucleus or a mitochondria or any other. Now, eukaryotes. U means true, and again, karyote, like before, refers to the nucleus. So the eukaryotes have a true nucleus, the one that we're more familiar with. They're much larger. They've got membrane-bound organelles like the mitochondria, the Golgi apparatus. They're far more complex, and they're far more recent. Evolutionarily, they're far more recent, and you can see here they have this nuclear envelope with these little pores in it. So all that chromosomal DNA is stored within that nucleus. Now, the origin of the mitochondria and the chloroplast are really interesting. It's thought that once upon a time, a eukaryote engulfed the prokaryote, and they formed a sort of endosymbiotic relationship where they helped each other. And this organism grew up within the cell, and they evolved together. And these prokaryotes gradually evolved into mitochondria and chloroplasts. Now that might sound a bit far-fetched, but there is evidence for it. And that evidence is that mitochondria and chloroplasts both have their own special DNA. That lends credence to the idea that they were once separate organisms. So there we are, a brief run through of prokaryotes and eukaryotes. Your turn now, so pause the video, have a little scribble out of this table and see if you can remember which of these organelles prokaryotes have and which eukaryotes have. And in a minute, the correct answers will flash up on the screen. Thanks for listening.